Hello, am I audible? Can someone please answer? Okay. Are my slides visible? Please let me know if my slides are visible. Anyone? Okay. Uh, if at any time if I'm not audible or if my slides are not visible, please let me know. Otherwise, I won't be seeing the chat box once I go into presentation mode. So someone has to speak on the mic and tell yes or no. Okay. We shall start now. Okay, so this is the week 3 live session for cell culture technologies, and I'm Sumati Raviraj, PhD student from University of Hyderabad, and the course instructor for this is Professor Manik Das from IIT Kanpur. So this week you might have watched the videos. Your syllabus included state-of-the-art facilities in cell culture lab 1 and 2, specialized facility in cell culture lab, interaction of cell and glass polycarbonate surface, and interaction of cell and glass polycarbonate 2. That is 1 and 2. So since the questions this time are little complex, so I didn't want any other introduction. We will start directly go to the question section. So as I said in the previous week, these questions I have made them uh, based on the assignment that you have. So if you are able to answer these questions, and I will also be explaining the questions and some related topics from the question. So if you're able to understand this and answer these, we will be able to solve your assignment easily. So let's go. So, but you have to answer the questions here. So you cannot like stay silent. I think very less participants are there this time. But whoever are there, please at least try to answer yes, no, you whether you know about it, you don't know about it. Please let me know because these live sessions are interactive sessions. So uh, few of you must talk on the mic. Okay, so let's start with the first question. uh, the dash lens in compound microscope produces the final image: objective, eyepiece, thin lens, thick lens. What is the answer? Anybody? I see at least 6 participants right now. I don't know if others will join later, but please try to answer the question. So no one is going to answer. At least say whether you don't know about it or anything. Just turn on the mic and speak, please. Okay. Okay, if you are not interacting, I will just go on with the slides. But please, if you know the answers at any moment, just start your mic and let me know the answer. So the first question was the dash lens in compound microscope produces the final image: objective, eyepiece, thin lens, thick lens. The answer is yes. Somebody was about to talk. Anyone? I even showed the answer now. You can at least tell me the answer. I heard someone's mic. Okay. Um, okay. So the first question's answer is B, that is eyepiece. So, so for this you have to know the basics of a microscope, a compound microscope. So this is the body of a microscope. This is the body tube. This is your eyepiece, and then it has adjustment knobs, coarse adjustment knob and fine adjustment knob. Then it has an inclination joint where this is fixed, and this is your base. And at the base you have a mirror, and this is where you'll get the light from the mirror. So the light source you can angle it using the mirror, and mostly the light for this kind of microscope is a natural light, so you will adjust the light using the mirror. And after that you have a condenser which will streamline or path your light to your sample. And then so this is your stage where you place the sample. You can see a holder sort of thing here. And then you have your objective lenses. So here is your point of magnification where you can adjust with different magnification lenses. That is 10x, 4x, 60x, 100x. And then you have your resolving nose piece. And then through here you will observe. This is your point of observation. Okay. So now let's see how the light passes through this, or how the image will form. So your object over here is here. You can see it is shown in angle of your microscope, however it is angled. So this is your eye. So your eyes will come here over your eyepiece, and then where you have your eye lens, and then you have your objective lens. These are the two important lenses that you need for a microscope. So your eyepiece lens is here and your objective lens is here. So this bigger lens is here, the smaller lens is over here, and then you have your object after your eyepiece, after your objective lens. So this is your stage where you will place your sample. So your sample is the object here. So how is your image formed? Does anyone want to talk? No? Okay. Uh, so this is how your image will be formed using the lenses. So this is your object. And where is your light source coming here? It's coming from here under the object. That is via your mirror and your through your condenser. So when the light falls on the object here, it will create an image because of the rays that are converging and that are diverging and then converging. Okay. So from here, your rays are converging and your image is formed here, and from here again your rays will converge through your object eyepiece lens. So your final image will be formed here from your eyepiece lens. So this is your initial image, which will be your uh, virtual image. So this is not your real image, this is your real image, and your initial image will be opposite or inverted through your object. Your object here you can see is like this, arrows downward, and your initial image is upwards, and then your final image will come here. Okay. So the magnifying power here you can know with your different lenses here, objective lenses. So your magnifying power formula for a simple microscope is given as m is equal to 1 plus d by f. So m is your magnification and d is your least distance of the distinct vision. Okay. and f is the focal length of the convex lens, focal length of the lens. So this is your simple working of your microscope. And then there are other microscopes as well. There are again inverted microscopes, confocals, and other microscopes where they use two or more than two lenses also. Going to the next question, which image can be produced on a screen? A real, B virtual, C all, D none. Anyone? Can you answer this? Anybody? Which image can be produced on a screen? Real image or virtual image? So just now I spoke about your real image and your virtual image. Your virtual image is before your lens where it is inverted, and then the real image is after your object, which is again upright, however your original object is. But in a microscope, when you put it, it is magnified. So your answer for this is a real image. A real image which you see through your eyepiece in your previous slide. The image which you see here, the final image, is your real image. And this image can be again produced on a screen for you to record it. Okay. So here you can see the difference between a real image and a virtual image. So this is your lens. Okay. So your real image will be formed like this, and this is your virtual image. This again depends on the type of lens you're using, or whether it is a mirror or a lens. So based on that, your the formation of your image will change. So location of your image, real images are formed on the opposite side of the lens, while virtual image are formed on the same side of the object. See, you can see here is your object. So your virtual image is on the same side of the object. And here you can see, because this is a one lens image, it is like this. So here you can see that. Just a minute. Okay, I hope my slides are visible and I'm audible. So as I was saying, the real images are formed on the opposite side of the lens, while the virtual images are formed on the same side of the object. So the real images can be projected onto a screen, but virtual images cannot. Formation of a real image. Real images are formed by converging of light rays. That is, this is your real image. So your rays are going like this into the cells. Here they are diverging, and then they converge to form a real image. Okay. And the virtual images are formed by diverging the light rays. So here your lens is here, your object is here. This is how the light is passing. So your from here your images formed by diverging rays. Okay. You should see from the lens how the light is passing. So here from the lens, if you see, the rays are converging. Here if you see from the lens, the rays are diverging out from the lens, and then at this point it is formed. Appearance of real images are always inverted, while virtual images are always erect. So here you can see your virtual image is erect, whereas your real image is inverted. Next, what is the units of osmolality? First is milliosmolar per kg, second is milliosmolar per liter, third is milliosmolar per kg, millimolal per kg, millimole per kg, and D is both A and C, milliosmol per kg, milliosmol per liter, millimole per kg, or both A and C, that is your milliosmol per kg and millimole per kg. Anybody wants to answer? Okay. So your answer is D. That is both milliosmol per kg and millimole per kg. Both are your units for osmolality, but more widely used unit is milliosmol per kg, because millimole will get confused with some other concentration units. So just to make it easier for osmolality, we use milliosmol more frequently per kg. So I just want to give you a comparison of what is osmolarity and osmolality. If you know the difference between molarity and molality, it's very similar. So in your osmolarity, it is the concentration of a solution that is expressed as osmoles of solute per liter of solution. That is nothing but your osmoles of solute in volume of your solution. Whereas osmolality is expressed as the total number of solute particles per kilogram. So here your uh, whatever per liter per kg per whatever you're seeing of your solvent, so that will also be in similar unit, that is weight. So this is, you can say, weight by volume, this is weight by weight. So this is uh, more accurate than your osmolarity, but osmolarity is more convenient for you to calculate something, or make a record of something. But if you want to be more accurate, if being accurate is your objective, then you should always go with osmolality. Why? Because uh, this osmolarity is calculated by volume of the solution, and this is by mass of the solution we discussed, and the unit for this is mole per liter, and this is osmol. This is a mistake, I guess. So this is all osmol per kg, not liter. Let me just change that right here. So this is your kg, not liter. Okay. So why is this more accurate than this? That is, why is osmolality more accurate than osmolarity? Because this also depends on some other factors like temperature and pressure, whereas your osmolality does not depend on your temperature and pressure. And also this will give you an idea of what amount is there in what amount in the same unit, so that is more accurate. So why this you have to know about this is, the concept of osmosis, where the water passes to the solutes. Okay, water always is attracted to the solutes, and it will try to maintain an iso tonic or isoosmolality. Okay, so here what happens when the osmolality is in hypo condition, that is a hypoosmolality. So in hypoosmolality, what happens if there are cells in that condition, in that environment? The water will enter into your cells, and your cells will start to swell, because in this case the solutes in the environment are lesser than the solutes in your cell. So that time the water will try to enter into the area wherever there are more solutes. Remember, here the solutes cannot pass through, that is why the water is passing to maintain the balance. So if it is hypoosmolality, your cells will get swollen and ruptured. If it is hyper, if it is the opposite, that is outside your solutes are more outside the cell, and inside your cell the solutes are less. So this time what happens, the water from inside your cell will go outside to maintain the balance. So that time what happens is your cells will dry out and they will shrink. Then isoosmolality is the water will be already balanced, neither outside is higher or lower in terms of solute concentration. So your cells will stay happy because the water exchange is not happening. There is no increase or decrease in water levels, it is maintaining it. So this is a measurement used for human fluids, so like urine or human serum. So for human serum, the normal range of osmolality for humans are from ranging 285 to 295 milliosmol per kg. So this is very important to maintain. If there is any change in your sodium levels or some other ion levels, then your hypothalamus will get signal to release more of your water content or send the water content to maintain the balance wherever there is higher salts. So now in your tissue culture media, the useful range of osmolality for cell culture media used for vertebrate cells are 260 to 320 osmol per kg. In children, the normal range of osmolality is 275 to 290. It is little lesser than adult human serum. Okay. Then osmolality can be measured by using an instrument called osmometer. So this is all about osmolality. Next, we can go into fourth question, which is in which microscope the cover slip, that is, the cover slip is something which you place on the sample. It's a small glass, square shaped or rectangle shaped or round shaped glass cover slip which you place on the sample to hold it in the position, so that when you put it in a microscope, if you want to go for higher magnification, it does not touch your sample and it does not move. Your sample will get locked inside it. Okay, so the question is in which microscope the cover slip is placed upright. Upright means facing to you. The options are A inverted, B upright, C both, or D none. What is the answer? Anybody? In which microscope, which type of microscope, the cover slip is placed upright? Anyone? Yes? Okay. Okay. When do you keep your sample upright? So the answer is B, in upright microscope. That is, see, this is how your microscope looks. Okay, both inverted microscope and upright microscope. So in your upright microscope, we'll first talk about this. So this is like the simple microscope that I showed you, the compound microscope where you get the light source from the bottom. So here also, if you see, so here also, if you see, your light source is coming from here. This is your light source and it comes from here. It will be adjusted over here, and then you have a condenser. This is your stage and this is where is your sample. Okay, this is your sample. So it is coming like this and it is hitting your sample. So your sample must be upright. So whatever sample is there, your light will pass through that. Okay, so because your sample will be either on your culture dish or on your slide. So your slide, bottom of your slide should face this light source. Then the light will pass through your sample and then it will pass to the objective lens. And from the objective lens, it will go here and this is your uh, eyepiece lens. So from your eyepiece lens, you can observe it over here. And this, there can be another another connector here. So from here, it will go into your camera and it will get recorded here. Okay. So this is your higher version of your compound microscope where you can record your image. So we saw that which image we can capture on the screen. So that is your final image which you get through your eyepiece, which forms somewhere here. So that image will be also recorded into your camera. Okay, so here if you see this light blue color over here is your slide, glass slide, and on top of that you have this pink color sample. Okay, so your sample, and this is your cover slip. One another small dash on top in blue color is your cover slip. So your cover slip is upside facing. So in your upright microscope, it will be placed upright. That is, your cover slip will be facing your eye, and the sample, which is on your glass slide, will be facing to the light source coming from the bottom through your condenser. And in inverted microscope, it is the opposite, because the light source in your inverted microscope, you can see here. So here I told you the direction, the light comes from here. This is your light source, it comes from here, it goes like this. Okay, that's why your bottom of your glass slide is facing the light source. So here, because your light source is on the top, so this is your light source. Wherever the yellow line is starting, that is your light source. So your light source is coming from here, and then it is going through the condenser. Your condenser will be here. So whatever was there here, it will be reverse here. You can see this is your condenser here. Same condenser is here at the top. Then this is your stage. Stage is in the middle for both. Excuse me. Then this is your objective lenses. Your objective lenses are on the top over here, and here it is at the bottom because it is inverted. So here, how will your sample be placed? So here your sample also will be placed inverted, so that the light source comes and hits your bottom of your dish, and then your sample, and then there is cover slip, and from the cover slip it goes through your objective lenses, and then from the objective lenses it will go to your eyepiece from where you can observe it, and then there will also be a camera for here which will also record it. So here is your adjusting knob, this is your eyepiece. So this is how you have to position your sample, based on what type of sample it is, based on uh, type the level of magnification that you do. Everything you should consider before placing your sample into the microscope. If it is a glass slide, always place it upright in the normal microscope and inverted in your inverted microscope, so you can understand, you can remember with the name itself. So first you should know whether your microscope is upright or inverted microscope. Just ask if it is an inverted microscope, then you can know how to place your slide. Okay, and also if your sample is in a culture dish, then what will you do? And if it's a live sample, if you want to do some live cell imaging, you want to observe your cells just for morphology sake, no need of live cell imaging also. If you just want to observe your cells, like in the previous sessions we saw that every time you split your cells or you seed your cells, you subculture your cells, you should always observe every set of subculture, every passage. You have to observe your plates, whether your cell is in proper morphology, if your cells are dying or if your cells are healthy, if there is any contamination. So that time you seed your cells in cover this cell culture dishes, right? So if you have an inverted microscope in your lab, you cannot invert your cell culture dish in that, because everything will spill out and your media will be gone, your cells will dry out. So that time you have to place it same only, you have to place it upright. So here in this image they have shown a culture dish. Okay, so in this culture dish, if you see, this is your cover slip. So they are growing the cells on the cover slip here instead of growing it on the dish. It's on the cover slip because glass cover slips help in better resolution for high power magnification and confocals. So you will always use glass bottom dishes when you're using confocal microscopes. So here when there is media, you can see, so you have to keep it upright, then your image will come via the light, it will hit here and it will come here. So in live cell imaging you do not have the option to invert, you have to always keep it upright. Other than live cell imaging, you have to fixed cells, it's called fixed sample. The samples can be either live samples or fixed sample. Fixed sample is you will dry it out uh, using either alcohol freezing, or you will use 4% paraformaldehyde, that is PFA. If you use that and fix the cells, you should always keep it upright on upright microscope, inverted in inverted microscope. Tribe cells will always be upright because you don't want to spill your media. Okay. Just give me a second. Okay. So we were talking about, yeah, we were talking about upright and inverted microscope. If anybody has a doubt, you can always ask me. Okay. So next question is, okay, so if you want to know more about your inverted microscopes and upright microscopes, you can go through the slides. I'll just read a couple of lines. Inverted popular is very popular with live cell imaging, and here you can observe the cells via the bottom of the cell culture, several, but there are also advantages over the upright microscope. So most cells naturally sink to the bottom of the vessel, whether it is adherent or semi-adherent. And if no suspension cells are used, suspension cells will usually float, so you have to fix them. But if they're adherent and semi-adherent, they will stick to your or settle down to your surface and adhere to your surface. So that time it's easy for your inverted microscope to check it in your media. And when growing in vessels that are suitable for inverted microscope, cells have access to larger amounts of media than if they were squeezed in between cover slip. So there is one more thing. You can also do live cell with cover slip, but it is not preferred because the cells will not get enough media that time. So here if you see in this case, so here if you are seeing in this case, that pink color liquid you see are also, it can be your media if you're doing especially imaging of bacterial suspensions. So that time what you do is you take like 1 or 2 microliters or some 10 microliters of your suspension, live cells of bacteria, and then you place a tiny drop on your glass slide, and then you place a cover slip on that. So that time your cells will still be suspended, but after a while the broth or your media will dry out. So this is not used preferred method for your um, live culture. So it's always preferred to use a glass bottom dish. Mostly glass bottom dish we do for mammalian cells because they are larger in size and you need the media in a cell culture vessel. This media within the cover slip is done for bacterial cells. In addition, sample access from top is not possible. Like I said, if you even try to, put it in an upright microscope and you try to, focus your cell culture dish in an upright microscope, if you place your petri dish or glass bottom dish here and your objective is coming here, so when you adjust your knob, your objective lens will move up and down. So based on your focal point, proper focal point, you will fix it on your particular plane. So that time what happens in low power magnification, there will be distance between your objective lens and your sample. But in higher magnifications, when you go for 60x, more than 60x, then your lens will start to touch your sample. It has to have that minimum to minimum gap to have that magnification. So that time, if your cell culture dish is on top of your stage and your objective is also on the top of your cell culture dish, and it is moving into your sample. So if it is a cover slip, it will move on to the cover slip and it will break your cover slip. Okay. And then if it is a live sample in like your culture dish, it will go and hit your lid of the culture dish. If you remove the lid of the culture dish, it will go and sink in the media. So for live cells, that is why we never do upright microscope. And another very important advantage is sterility. As there is no contact between the objective and the sample, sterile working conditions are guaranteed. So just now, as I said, when you are going for high power magnification, your objective will tend to touch your sample or your cover slip or the bottom of your dish. So when it is touching your sample over your cover slip, or if it is sinking into the media, you can ask what happens if it sinks. But again, there will be again different reasons. One is, first thing is you will lose your sterile culture, and the second thing is the refractive index of your media will be different. You need oil immersion for your high power magnification. So all these things come into play. So when you're doing it in your inverted microscope, the objective is coming from the bottom and it will hit your glass bottom plate. So that time what happens, that time what happens is your your petri dish, you can parafilm it also, so it will not be contaminated with outside exposure to anything like your objective lens or your air medium. And there is also possible to access your cell culture dish for exchange of media, or adding some drugs, adding some toxins, and checking the immediate effect of your addition to the medium. So when you're doing like this, if you see here, this is your cell culture dish. So here, usually when you're doing live cell imaging, there will be a rectangle box in which they will place your cell culture glass bottom dish. So in that rectangular box, there will be a provision to maintain the 37-degree temperature which is required for the healthy condition of the cell. And there will also be an option to supply CO2, 5% CO2 to maintain the pH. So you may also not have to worry that if you're doing live cell imaging for more than like 15-20 minutes, if it's a longer time exposure, you want to check something which is happening over a period of 30 minutes or 1 hour, and you don't have to worry that your cells will die because of temperature difference, or it will die because of, or it will have some other kind of activity because of increase in acidic nature in the pH, because there is no carbon dioxide to maintain it. So this live cell chambers, they have this option for pumping of 5% CO2 and maintaining 37 degrees temperature. So that time what happens, your cells will be healthy and you can capture your changes in your cell. And you can also, there will also be ports in the live cell chamber. There will be some option to pipette either fresh media or remove little media, or you can also pipette in your, or inject your drug into your cell or into the media. You can inject some kind of hormone, you can inject any kind of cell arresting material if you want to see if this uh, chemical is able to stop the proliferation of your dividing cells, something like that. Whatever you want, some external treatment you want to give within your live cell and see the immediate change, you can always add it via your cell culture chamber. Okay, this live cell chamber. So that is your advantage of your inverted microscopes. That is the reason most of the high power microscopes, confocal microscopes, live cell imaging, high content microscope, all these microscopes will be inverted so that they can have all these provisions for it. Uh, Next is upright microscope. So what is the advantage of upright microscope is you can use upright microscopes for phase contrast and wide field fluorescence. So in wide field fluorescence also you can use upright microscope. Only from confocal and other things you need inverted. So here the life sensor samples that are squeezed between the slide and the cover slip. So even here, as I said you, the bacterial cells which you can see live, you can use in upright microscopes because it will go and touch your cover slip. But even there, if the focus is not proper, if you were, trying to focus more, then you will break your cover slip also. So mostly this is done with fixed cells, and in principle upright microscopes can be used for live cell imaging, especially with dipping objective lens. The system is often used for larger samples like tissues and animals. Okay, so when you do microscopy for animals, like you can do the intravital microscopy, that is nothing but you dissect the animal. It is live, it is under anesthesia, you dissect it. If you want to focus on particular organ of the uh, animal model, that is either your mice or whatever you use. So that time what happens, you will have this dipping objective lens which can go inside the body of the model organism, that is your model animal. So that time also we use upright microscopes, and working distance of upright microscopes are longer generally because you don't want it to hit your sample pretty soon, which but this also leads to lower resolution and weak fluorescence signal. So your fluorescence signal will have point spread function, so it will disperse. You will not have a very high resolution image. Because of this, your light will get scattered and dispersed. In contrast to inverted microscope, upright microscope do not allow for the use of objectives with high numerical aperture directly on the cover slip bottom on which the cells are there. So obviously in upright microscopes, if you use high objective lenses, so high objective lenses will have higher numerical value, they will have higher magnification, so they have to go more closer to your sample. So it is not possible in upright microscopes. Therefore, we recommend inverted microscopes for live cell imaging, as I said. So this is your advantages for both inverted and your upright microscope. So the next is, since you all are not answering, let's just go like this. So your next question is, glass or plastic surface which is used to grow which type of cells? A glass plate or a plastic plate, which types of cells can you grow on this surface? Anchorage dependent, anchorage independent, monolayer, or histotypic? Anybody? Anyone wants to answer? I hope you all are listening. Okay. So your answer for this is A, anchorage dependent cells. So you generally grow your anchorage dependent cells. If you remember in the previous sessions, I showed you different types of cell culture dishes, your T25 flasks, your T75 flasks, your 100 dishes, 60 dishes, 35 dishes, 24 well plate, 12 well plate, 96 well plate. So all these are made up of plastic or glass, mostly plastic. It's called polystyrene plastic, which is generally used for tissue culture or cell culture. And like I said, if you want to do your confocal microscopy or high power microscopy, then you will go for glass bottom dishes. So in this glass bottom dishes, there will be this central area which is made up of glass. So that will have proper refractive index and thickness for your high resolution images. So your anchorage dependent cells are nothing but your adherent cells. So they need a surface to attach. So they will attach in most of your surfaces. And your anchorage independent cells, it doesn't matter whether you use glass or plastic surface, they are in cell suspension state. So if you just give them suspended media, they will grow in that. Your monolayer cells, so your monolayer cells also now can be grown on your glass or plastic surface. But here what you have to remember is, when you want to grow them in a proper monolayer, your cells must be grown in a plate which is treated, either tissue treated, or some tissue culture anchorage uh, treated, or some polylysine treated, something like that. And if you want to grow monolayer, means many things, it's not only anchorage dependent, you can also grow anchorage independent cells on your monolayer. If you just, if you don't want to subculture or proliferate them, if you want to fix them, how will you do? So then also you need a monolayer for your fixation. So when you fix your cells on the plate also, or on the cover slip, you cannot overlap your cells. They should be in a single layer. And then there is histotypic cell culture. So your histotypic cell culture is your this tissue cell culture, or your organoid cell culture, or a biopsy, or a primary, which you grow in a tissue, as a tissue. So why the surfaces are important? There is again surface characteristics which you should know when you're doing different types of cell culture, and for different cells. Firstly, you should know what is the chemistry of the cell. That is, whether it is glass or plastic, or whether it is coated or whether it is not coated. Does it have lysine? All these things. So that is your chemistry. So which type of cell needs what, that will be given by the cell culture in their catalog, from the source you get it. And then there is wettability of the surface. Wettability of the surface is how hydrophobic or how hydrophilic the surface is. So because your cells also have your cells have charge, and then the surface also will have charge. Your hydrophobic and hydrophobicity is dependent on the energy of that surface. So if your cells are getting repelled by your surface, then there is no point of using that surface if you want to grow an anchorage dependent cell. So wettability of a surface is whether your surface is hydrophobic or hydrophilic. If it's hydrophobic and hydrophilic, how hydrophobic, how hydrophilic. So you can even change your intensity of hydrophobicity, hydrophilicity, based on different coatings, based on different hydrogels that are available. Okay, so generally anchorage dependent cells will not prefer hydrophobic surfaces, because it repels the cell. And your hydrophilic surface will be helpful in attachment of the cell, adhesion of the cell on the surface, and it will help. Once it attaches, it will help in its growth and proliferation. Whereas your suspension cells, you can grow in the hydrophobic plates. So your hydrophobic plates you can use for many reasons. If your experiment needs it, like if you don't want any, contamination of other sources in your surface, or you don't want your cells to sink and the settle on your surface. You want them always suspended form. Then you can use your hydrophobic plates. And then there is stiffness. So your stiffness is depending on, like if you want to do biophysical characterization of the cells, like you want to see the tension, the tensile force the cell exerts. The cell has the cytoskeleton which helps it to adhere, and then adherent cells, they also move, they migrate on the plate. If you want to see the traction force, it is called, the cytoskeleton exerts on the surface, that you can measure when you coat your surfaces with a different with uh, with some coat like gelatin or collagen with different stiffness. Like there are hydrogels and other gel based coatings that are available, which have different degree of stiffness. So based on how much stiffness you want to give to the cell, because your cells in your body, they attach at different places, right? Like few cells will attach to your bones, or few cells will attach to other organs. Few cells will be attaching to each other in a, specific uh, tensile strength. Like your muscle cells or your heart cells, they need a specific strength for them. They always have this specific tension that they have, because they contract and they relax. Other cells may not need that tensile strength. So when you want to have that kind of pressure on your cell, and you want to measure that tensile strength of your cell for different environments or different media, types of media, different treatments, and that time you will use this gels that is of different stiffness. And then based on the level of stiffness also, you can see how your cell morphology is differing. Not only morphology, uh, if your stiffness varies, the activity of the cell also will be different for few cells. Like they might be more active at certain stiffness, they might produce more ATP at certain stiffness, they might need less energy for certain stiffness. So you can measure all these things using this stiffness based coatings. And then the topography of the plate. Now if you want to grow just a mono layer of cells, then you will just need a flat surface to grow on it. But then if you want to grow like different types of culturing, like co-culturing two different types of cells, or you want to see like migration of the cells from one cell area to other cell area, or you want to do 3D culturing, 3D co-culturing, organoid sphere, 3D sphere culturing. So that time you need your topography of the plate also to be different. It will either be, it will have a depth in it, it will not be flat, or it will have some kind of partitions in it for different layers for different types of cells when you co-culture. So topographic also comes into play. Now how the cell responds to this. So if you see, depending on the surface, you know how your cell will spread and grow. Like I said, this chemistry of the cell plate, and your wettability of the cell, wettability of the plate, your stiffness will play a major role in how much your cell will spread over the surface. Your cells will have a particular morphology. So when they are suspended in liquid, any cell will be spherical. So when they go and attach, they slowly will start to spread their cytoskeleton, have that tensile strength and pressure, and they will start to spread. So depending on all these things, how much uh, the percentage of everything is, your cell will spread to according to that, relative to that. Then your cell detachment and cell death. So if these things are not favorable to your type of cell, then your cell may not attach. It will just stay like this, like a sphere on the plate. But if they are not suspension cell lines, then they will slowly enter into cell death, because it needs attachment for its growth and proliferation. So if it doesn't get attached, it will eventually die. So that's why this is all important. So here you can see like few examples I have given, like organ culture, explant culture, and then there is dissociated cell culture, there is organotypic cell culture. So you can see here how the plates are different for each. So in your organ culture, there is partition, you have this kind of depth here and all. And then in your explant culture, you have this kind of surface where your tissue and the solid liquid interface can be made for the cells to migrate. So it's and it will form some kind of outer growth or something like that. And then there is your other type of plate, which is called as dissociated cell culture. So here you can see that sometimes they might disaggregate, or the cells from a monolayer at a solid to liquid interface will be at a monolayer. So it's like that. And then here in organotypic, you have a completely different kind of plate, which you can use for co-culturing, because in organs you have different types of cells, and you will not just maintain one type. So that time you see you can have compartments, and then you can sandwich that with another layer of a coating of something or a hydrogel, and on that you can coat another layer of different cells, 2 3 layers you can coat. So you have different equipments for different type of cell culture. So, cell culture dishes can be coated with a variety of materials, including extracellular matrix proteins. So, extracellular matrix proteins help in adherence of your cells. So, and they also can be used to coat your cell culture dishes to regulate the differentiation and proliferation of the cells. So, your extracellular matrix will also have proteins which are secreted by other cells, or inbuilt proteins in your matrix based on where you get your ECM from. If you're coating it by yourself, then it will have a component that it has so and so proteins. So based on that, you can buy different ECM coatings and you can coat it on your plate. Or else if you're growing this in like co-culture or even a monolayer, everywhere the cells secrete extracellular proteins. So within those also, depending on the media or depending upon the stimulus you're giving, they will secrete different proteins. So this might help in the differentiation, proliferation and all. Like in the previously I said, like we uh, differentiated cells, we transfect it with SV40 and then we proliferate them in 33. So why that is happening is because in 33 it is producing certain proteins which help the cells to proliferate. So the proteins will be produced within the cells, and then it will also be released, and then it will stimulate the other cells surrounding to it, and then they proliferate. And then when they are put into 37 again, they stop producing these proteins. Now the proliferation will stop. Then now, because this condition is favorable for the differentiation, they will start to differentiating. So like that you make use of ECM proteins. And then cell based membrane gels, like I said, the hydrogels and all. The gel can be used to coat the cell culture dishes to provide a surface for attachment of induced pluripotent stem cells. So this is done usually in IPC cell cultures, where you give a surface to the cell uh, which will be useful for induction of your pluripotent stem cells. Then you can coat with polylysine, like I told. Like if you have semi-adherent cells like HEK cells, they tend to detach from the plate. Generally they will attach, but then easily they will detach from the plate. So to make it more adherent, we can coat with polylysine, and this will help in mediating negative charges, because polylysine will have negative charges of the cell membrane and the negative charge of the surface. Then we can also coat with polydopamine and collagen. These materials can be used to coat micrograted surfaces. So there are again different surfaces you can see, like here, and then this type of topography. So for different materials can be coated at different surfaces. uh, then laminin and collagen. These materials can be used to coat normals, any type of cell culture dishes. Laminin and collagen, they are help in attaching the cells, and it will help in maintenance of the structure, proper morphology of the cell. It will help in its cell spreading. Then Corning is a company. So you not only Corning, you can use any other well sophisticated company. They will provide matrigel matrix. So there are very few companies which provide this. So this material, the cell culture dishes are often made with clear plastic or borosilicate glass, so that microscopic observations can be made without distortion. So as I said, the clear plastic or borosilicate glass, the cover slips are always glass cover slip, that is borosilicate glass. They have this proper thickness, and they have proper clarity for resolution, better resolution, without any distortion in the observation of the sample. And the plastic is the polystyrene plastic that we use. Usually if you go with like low power magnification, polystyrene glass is plastic is enough. It does not give much of your distortions, but when you go to high power magnification, you need your glass cover slips or glass bottom dishes. So next is Next question is the ability of the objective to capture the incoming light. What is this called? Is it called wavelength of incident light on lens? Is it called optical resolution? Is it called numerical aperture? Or is it called focal length of lens? What is it called? The ability of the objective to capture the light. Anybody wants to answer? There is a message. Yeah, somebody wants to answer. I hear a mic turning on. No? You can answer if you know, or if you don't know anything, you can just tell about it. Okay, so
Numerical aperture.
yes, yes, very good. Good, at least someone is answering. Yeah, so the ability of an objective to capture your incoming light is called as numerical aperture. So your numerical aperture is this cone that is formed between your optical axis. Okay, the point of your optical axis, like that is nothing but the center of your sample to the rays that come out of it, light rays that come out of it, and the maximum light that reaches the lens, objective lens. Like you see this semicircle, right? So the semicircle is the entire light that can get reflected out of that sample. But you can see out of this entire semicircle, only this triangular area is being able to enter your lens. So this ability of your objective lens to capture the maximum possible light that is coming from your sample is called the numerical aperture, and this has been already calculated, and this does not have any unit like for magnification x value, or you have for distance some centimeter. Numerical aperture does not have any unit. So what is numerical aperture? It is the most important number that is associated with the light gathering ability of an objective or a condenser, and it is unitless or dimensionless. So it is directly related to the angle of the cone. You see, this is your cone that is being formed. So angle of the cone which is being formed from the point on the specimen. So this is your, where's the cursor? This is your point on the specimen. This is your point on the specimen, and this is your cone that is being formed from the point. And then the front of the lens of the objective or condenser, this is your objective or your condenser, whatever, whichever is first. It depends on the type of microscope. So this cone that is formed, and the angle you see from this point, whatever point is at the center, that is your optical axis. This black line you see is your optical axis. So now from this axis, if you go this side, you will get an angle till the end of, that is nothing but your alpha or theta. Few of the literatures or few of the books you can see that it is defined as alpha, and few of the places it is also defined as theta. So don't get confused, both are the same. So the formula that has been derived for it is n sine theta or n sine alpha. Usually it is this angle. Theta is your angle, and then your sine theta. You should know this, your cos, sine, tan for this. And n is nothing but your lens. Uh, in Yeah. So then you have different NA values for your different magnification of lenses. So your 4x lens, that is your least magnification, that has a NA value of 0.1, and then you have your 10x, that is your 0.25. So if you see your objective here, your objective lens, so whenever if you get access to a microscope, even a simple compound microscope, if you have the access to it, you always check the objectives. Like many of the information will be written on the microscope itself. You need not, study it from your books or something. If you just look at different parts of your uh, microscope, you can see that there is a lot of information that the product holds on itself. So in the objectives you see, every objective will have certain numbers written on it. But you should know what that number is. Few of the objective lenses will also write it, like NA is 0.25, this is 0.2, minimum focal distance is so and so. So it will be written. But maybe in few of the lenses it would be written. But if you get to know what everything is, then you if next if you're checking any other objective, you will remember, okay, this value corresponds to so and so terminology. So like that you should always check the objective. So here you see, here it is a 10x microscope. So it's written 10 first. So that means the magnification power of this microscope is 10x. And then it has also written that it is 0.25. So 0.25 is any value. Usually in recent microscopes they write is at 10x. So if they have written it as 10x, you can understand that it is magnification, because x we use for magnification. And then beside that, if there is some 0 point value or 1 point value, that is the numerical aperture of that. And then uh, what else is written, some 160 by 0.17. So these are all different things that they write. Like in the next you can see that 40x objective has a numerical aperture of 0.65, and then 100x has a numerical aperture of 1.25. So there are increasing, as you see the magnification is increasing, you can see that the numerical aperture value also increases. So more the numerical aperture, more light it can gather. You can see, right? If the aperture is more, the cone will be more wide, and then more light it can gather because of the wider cone, and then you will get more information from it. More light means more information. That's it. So higher your magnification, higher will be your uh, numerical aperture. And then they also have something like 160 written, and then they have 0.17 written. So here in this case, if you see, 0.17 is nothing but the thickness of the surface that you're using, either your plate or your your cover slips. Generally your cover slips will be made of 0.17 to 0.18 or 19 thickness. That will be your thickness of the cover slips. So they are saying that this objective lens can magnify very clearly if the thickness of your cover slip is 0.17. So you can know that for what objective lens what thickness you can use, because they have given it already. And then few will have some other like WD, that is like working distance, like how far the length should be. Usually the recent ones do not have this. This is one of like the limitation or something of few lenses. And then some other properties, it will tell you whether you have to use oil immersion or you have to use air itself is enough. If you have higher power, like maybe from 40x or above 40x, maybe from 60x, 40x to 60x itself, you can start using oil. So as your Na value increases, your refractive value changes. To balance that, you need to use oil immersion. Okay, so all this you can see. And then what else? That's it for here. Next question is, name the instrument to measure osmolality. We just discussed what is osmolality and osmolarity. So what is the instrument that we use to discuss to measure osmolality? I have also explained this in the uh, question there. I told you what is the name of the instrument. Anybody wants to answer? The previous question where we did osmolarity and osmolality discussion, there I told you the instrument. What is the name of the instrument that we use to measure osmolality? Anybody wants to answer? Let me know. Where is that question? See this one. Here I mentioned what is the instrument that we use to measure osmolality.
meter,
Yes, sorry, once again.
osmometer, I think.
Yes, yes, correct. It is called osmometer. And one more thing here I forgot to say, the small n. So the Na which I was talking was your short form for your numerical aperture. The n, small n here is the refractive index. That's what we were talking, right? The oil and air. So Na, small n is the refractive index of the medium in whichever you're doing. So if you're doing in lower magnification, uh, air is your medium. So you will use the value of refractive index of air. If you're doing in higher magnification oil, you will use the refractive index of oil. Now recently there are newer microscopes which have also come, which use water as your immersion. So there you will use water as a value of water, refractive index as n. So yeah, so someone answered here, very good. So the answer is osmometer. It is not osmometry. Osmometry is the principle of it. Osmeter is not the answer. Osmometer is the answer. Osmometer. Okay. So this is how a osmometer looks like. Then there are different types by which you can measure the osmolality. There are different principles. Okay. Based on the different principles, you can have different instruments also. One can use this, one can use this. So it's different. So one is freezing point depression is one way how you measure your osmolality. The other way is membrane osmometer. So this is a different osmometer. For freezing point, you'll use a different osmometer. For membranous osmolality, you will use a different osmometer. Then there is vapor pressure osmometry. Then there is boiling point elevation osmometry. Okay, these are the four types. So the first, what is boiling point elevation? So it is not useful in clinical and live samples, or cellular kind of samples, because it coagulates your proteins, because it's boiling point, as the name suggests. It will, you have to boil the sample to a temperature where it reaches its boiling point. You have to heat it that much. So that time your cell will die, your proteins will coagulate or aggregate. So mostly we don't use this. The the second is freezing point depression osmometer. So the freezing point depression osmometer of a solution is related to the osmotic concentration. That is why we take this principle and we use it in the measurement. So it is a difference in the freezing point of the solution without the solute and with the solute. It's like that. So if the concentration of the solute is increased, it will lower its freezing point. Obviously, if there is like pure water or something, its freezing point will be at a particular temperature. And then if you add some solute in it, then the freezing point will get lowered. So normal pure water freezing point is minus 0.53 centigrade. At minus 0.53, the normal pure water will freeze. But then when you add solutes in it, depending on the amount of solute or depending on the types of solute it has, depending on the population of solutes, the freezing point will change. So you can use this, this instrument uses this difference in the freezing point to calculate the osmolality of that solution. Most commonly used method for measuring the osmolality of serum, this freezing point depression is the most common, because when you freeze, it will not harm the cells or it will not harm the protein. It will just because we also use freezing to store certain things, right? So freezing will not harm it. So that is why this is most widely used. And the third is vapor pressure osmometer, measurement related to the decrease in dew point of the temperature. This is freezing point, this is boiling point, this is dew point. of pure solvent caused by decrease in vapor pressure of the solvent by the solute. So dew point will be a temperature for pure solvent, like how there was a particular degree for freezing point. In the same way, there will be a value for dew point temperature of a pure solvent. This is a, and then there is, when there is a solute, there will be decrease in the vapor pressure. So dew, when is the dew formed? When it's heated and then it forms, it condenses and then vaporizes and then it forms this dew drops, right? In the morning we get this mist and dew drops, so it is that concept. So when it vaporizes and then condenses back and it forms a dew drop, the decrease in the vapor pressure of the solvent because of the solute is measured. Same principle like with the solute. Without this solute, what is the difference? One drawback of this is uh, the measurement of volatile solute in the serum is not better, because if it is volatile, volatile means what? It has its own property to vaporize. So measurement of any volatile solute is not possible in the serum. Volatile gases, if present, will increase the vapor pressure of the solvent, so it does not work for them. So this is not clinically advised, because there are many volatile things also in the samples we measure. The other one is membranous osmometer. So here the device is used to indirectly measure the number of average molecular weight of a polymer sample. Okay, so it will measure the average molecular weight. Then one chamber contains pure solvent and the other a solution in which the solute polymer with unknown molecular weight. So same thing, everywhere you will do a pure solvent and then your unknown, and then you will see the difference, that the machine will see the difference and it will give the value of osmolality. If you want to read more about it, if you want to know, in detail the working principle and the mechanism of this, I have given the slide share from where I have taken this information. You can always visit that. It has a very detailed description. I don't want to get into that much details right here, because the main point was to let you know the different types of osmometer and the principle behind it. Okay, so the next question is, which surface helps in cell growth and proliferation of skeletal muscle cell? A. Polystyrene, B. Hydrophilic coating, C. Suspension dish, and D. Hydrophobic coating. Anyone wants to answer? Which surface helps the cell growth and proliferation of the skeletal muscle cells? You know what type of cells, right? Skeletal muscle cells are, whether they are adherent or they are non-adherent, they are anchorage dependent or independent. So based on the type, based on the knowledge of the type of the cell, you can guess what kind will they prefer. Will they prefer polystyrene, that is your plastic, or will they prefer some hydrophilic coating or hydrophobic coating or suspension dish? How what is your answer for this? Anyone wants to answer? Anybody? I told you I won't be seeing the chat very often, so I don't know A is for which question. Maybe it's not for this because it's some 3-4 minutes earlier. So please open the, turn on your mics and you can answer the question. Which surface will your skeletal muscle cells like to grow on? Okay. So
Hydrophilic coating. Yes.
yeah, so your answer is hydrophilic coating. So you can also grow them on polystyrene, but hydrophilic coating will enhance its adhesion, so that will help the cells to attach and spread and properly grow and proliferate on the surface. So in vitro, muscle cells prefer hydrophilic surfaces over hydrophobic ones, especially for long term viability and proliferation. Hydrophobic surfaces reduce surface wettability. Like we spoke previously, wettability is the nature of it to hold the liquid, aqueous liquids, or uh, the 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 percentage of it that is hydrophobic or hydrophilic, that is wettability, which inhibits mammalian cell adhesion. So if it is hydrophobic, it will inhibit the mammalian cell adhesion. It will repel the cells, like I told you before. If it's hydrophobic, it will repel your cells. So that time your cells can't go and attach properly. So there are some examples like hydrophilic materials, more fibroblasts can adhere, and then it can spread on more hydrophilic polymer surfaces, like your hydrogels are there, your pectin gel hydrogels are there. So these are all your hydrophilic polymers or your hydrophilic surfaces. And then protein absorption, slightly hydrophilic surfaces have better protein absorption and the cell adhesion properties than your hydrophobic surfaces. Obviously, if they attach more, they will be able to take up your proteins more. Then spatial conformation, the conformation of the absorption molecules may mediate cell adhesion rather than number of ECM molecules. So this is the thing, like when it spreads out, so the spatial conformation of the molecules around the cell will help in mediating it. It's not like you have a plate and then there are a lot of ECM molecules in that, but your cell is not adhering. So it doesn't mean like just because ECM is there, it will adhere. The ECM has to be spread properly, it has to be coated evenly. If it's like a lump somewhere in your dish at some corner and your cells are here somewhere, then it will not help your cell, right? Your cells can't take up that protein and attach. So the spatial conformation of the protein, how well it is spread on the surface, also helps in attachment of the cells to the plate. So here you can see in this picture the skeletal SC's isolation from skeletal muscle biopsies. So skeletal cells, you can see they are isolated and then they are plated onto such kind of dishes with some coating. Even if you don't coat, they will adhere, but the growth will be slow and they might not proliferate. That is the problem. So it's always better to coat the cells. Usually we have this TC treated plates that have some kind of coating for the adherent cells. So when you seed them on such plates, they will nicely attach and then they will amplify or proliferate in that. And then from there you can then again even go to different kind of plates with different coatings on it or different attachment surface on it to make a different type of model from it. So if you grow on plates like this, you will get this monolayer of cells. But if you want to go 3D or scaffold models, then you can go to some cell sheets, scaffold free model sheets, and myoids, and then you can go for this scaffold models hydrogel. So this will help in different arrangement of your cells and will give you a different type of uh, tissue if you want to check something at a tissue level. Okay, so next is, if the water drop forms an angle less than 90 degrees with the surface, then the surface is what type? A, hydrophobic, B, neutral, C, hydrophilic, D, it cannot be determined. Anybody? Let's answer, come on, somebody answer. Yes, if the water drop forms an angle that is less than 90 degrees with its surface, then what type of surface is it? Is it hydrophobic surface? Is it neutral? Is it hydrophilic? Or like, can you not determine it based on the information that is given on the question? Somebody speak up. Anyone? Come on. You can just guess, right? Like there is a water drop on the surface. The angle that the water drop forms, that means if there is one drop, then from the surface of the drop, to surface of your base, what is that angle? If that angle is less than 90 degrees, how much will that water spread on the surface? That means, did it like the surface, did it not like the surface, or it doesn't care about the angle? What is it? Anybody?
Hydrophobic, I think.
Hydrophobic. If it is less than 90 degrees with the surface, the angle. Okay, okay, at least someone spoke up. So good for trying, but the answer is hydrophilic. So maybe it was, I didn't, help you in imagining. So let's see here in the picture. See, what did I say? If you drop a liquid on the surface, the angle between the edge of your drop and the surface. So this is your angle. So this angle is less than 90 degrees, right? This is your center point. This is your less than 90 degrees. Then your liquid will spread. So that means there is less space between the liquid and your surface. So it's trying to, go and spread on it. That means the surface is hydrophilic. So it's water loving. Hydrophilic means water loving. So it is uh, this angle is called as contact angle, based on which you can determine the hydrophobicity of your surface. If the droplet spreads on the surface, wetting a large area of the surface, then the contact angle is less than 90 degrees. Like they said in the question, it is less than 90 degrees. Then that surface is considered to be hydrophilic or water loving. If the water drop forms an angle greater than 90 degrees with the surface, then the surface is hydrophobic. The water tends to bead up on the hydrophobic. Like generally, if there is some kind of oil or some, some kind of surface where the water doesn't, stagnate, or it doesn't, spread, it doesn't become like this layer of water, it just stands like a round bead on top. You might have observed surfaces like that, right? Like this Teflon surfaces, or waterproof, water resistant surfaces, your glasses on your watches, which are water proof and water resistant. There, how the liquid is, it will stand like a bead like this, right? Then how this angle is, have you noticed? If it's like a circle here, your angle will be more, right? It won't be as low as this angle. So then that means that much, that is more hydrophobic. So if the angle is like this, then it is highly hydrophobic. It's like 90 degrees or little more than 90 degrees also, it's hydrophobic. But it is lesser than 90 degrees, it is hydrophilic. So here you can see my explanation how it gives. So here you see your contact angle. This is your surface angle, and this is this is your straight line. You should imagine your straight line here. That is your 90 degrees. So here you see this angle. This angle is less than 90 degrees. Your straight line is here. Your angle is less than 90 degrees. So it's hydrophilic. Here you see it's more like a bead. It's like a, not a perfect circle, but still a circle, half circle, little more than half circle. So here you see your angle is more than 90 degrees. This is your center line again. Your angle is more than 90 degrees between your surface and your this edge of the liquid. This is the edge of the liquid, and this is your surface. So you see that angle between that, this is your angle. So this is more than 90 degrees. That means it is hydrophobic. And then there are super hydrophobic surfaces like this nano materials or some chemical technology materials which want, like, the you might have recently seen in these ads that they have this waterproof or water resistant clothes, that even if you pour a cup of water or something on it, the water will not get absorbed, it will just flow away, the cloth will always be dry. So those are super hydrophobics. So this is the concept that they use to make such products. So these products will, when the water falls on it, it will have a angle that is greater than 150 degrees. Forget 90 degrees, it will have greater than 150 degrees. So the water will not even stay on that surface for long. It will just roll away from that surface. So that is called as super hydrophobic. So this is important for the wettability. So same thing, your cell also has water content, right? So the surface has to go and attach. If your surface is so hydrophobic that your cell can't go and, even sit on it properly, then it won't attach. It will just, roll away. It will die after few days. Okay, so this is important for your wettability and your degree of attachment of your cell to the surface. So this is the angle with which you determine that, and this is called as contact angle. Okay. Your last question. Which of these are anchorage dependent cell uh, cell cultures or cell lines? A. Hematopoietic cell lines. B. Cardiac cell lines. C. Skeletal muscle cell lines. And D. Neuronal cell lines. We already discussed about adherent and suspension cell lines in our previous sessions. Hope you have watched the video or attended the session. So this was discussed in that. Hematopoietic, cardiac, skeletal muscle, neuronal. Out of these, which is the anchorage independent? Anchorage independent means suspension cells. Answer. Anyone? Okay, think. You know, na, cardiac cells are what? Heart cells. Skeletal muscle are your muscle cells. Neuronal are your brain, neurons. Hematopoietic cells. What are hematopoietic cells? You might have heard hematopoietic stem cells. Means what? Your cells from your blood. So these are cells which are in your blood, which is suspension, which is adherent. Okay, nobody wants to answer. So your answer is A, hematopoietic cells. These are the anchorage independent. That's why they are in suspension. They are in your blood. So it's in fluid. They don't attach anywhere. They just go wherever your blood flows. So here you can see again like a revision. There is adherent cell culture. There is suspension cell culture. In adherent, you can see that it is attached to a solid or a semi-solid substrate. Then it is easy visual inspection because it's at one place, it's stagnant. Cells are dissociated enzymatically using trypsin. uh, Growth is limited by surface area. No agitation is required. It's used for many applications like cytology, harvesting it for western blots or RT-PCR, cloning, and then many other research applications. Uh, on the other hand, suspension cell lines, they are grown floating in a cell culture medium. They require daily cell counts, viability determination. So when you do this suspension, you have to be sure that maximum number of your cells are alive, because your dead cells also will float. How will you differentiate? You have to keep a count of your live and dead cells here. In adherent cells, if your cells die, they detach and they float. Then if you change your media, they will go away. But in suspension cells, that is not the case. You have to centrifuge and take all your cells together. You have to resuspend all your cells together. So you should always see that you are not letting your cells die. Okay. And then it does not require, one advantage for this over that is it does not require enzymatic or mechanical dissociation. So you don't need agents like trypsin or any other cleaving enzymes. Then growth is limited by concentration of cells. And so the limitation is there. How much growth is limited by surface area in other end cell means how much surface area you give for the cell will grow that much. Grow means by in terms of proliferation, and also in terms of the spreading on the surface. The more surface you give, it will spread more, it will proliferate more. And here growth is limited by concentration of cells. Like you have a particular volume of your media, Now, number of cells, it will reach a particular percentage, like some 80% or 90%. If there is no more place for your cells to get suspended freely, and keep moving in the media, keep floating in the media, if they are overpopulated, then that is a limitation. Then again, you have to split them. Next is it requires agitation. Yeah, see, like I said, it needs to be moving and it needs to be floating. So it needs the constant stirring or shaking, like you do for your bacterial cultures, which are cell cultures, which you do in broth media. You keep them in rotation, right? You keep them in this shaker incubators. So because whatever it's producing, it is dissolved in the media. So if it's some carbon dioxide it wants to release, or some other gas it wants to release, that gas will be in the media broth itself. It won't get exchanged easily because the cells are also there. So if it's constantly getting stirred, all this exchange will happen, and also for maintaining the pH and all these things. Okay. uh, pH in case of mammalian cells. Then it is used for bulk protein production. So this is one advantage here, is because you don't need much surface area, you only need more volume here. So in a given volume you can produce number of cells, much more number of cells than adherent cells. So because of that you will get more value, more amount of proteins in the same space, because you don't need only that area where it needs to spread. You need how much media you give, it will grow that much. So you will get more bulk amount. So this huge protein production companies or bulk production labs which do this very large scale cell culture, they have this huge incubators where they grow the suspension cells, and they have this constant media monitoring, cell health monitoring, exchange of media. So they get like huge amount of proteins from that. And then they are harvested in batches, and then they are used for different research applications, like for cloning or recombinant technology, etc. Anchorage dependent is nothing but adherent or monolayer, and it must be cultured while it is attached on a solid or a semi-solid surface. So if solid surface, again becomes like your glass or your polystyrene, which is coated with some hydrophilic substance, or your semi-solid is your hydrogels or your pectin gels, your collagen. All these are your semi-solid substrates. So example for your anchorage dependent cell lines in vitro are MCF and PC3. And MCF is your breast cancer cell line, and PC3 is your prostate cancer cell line. Then anchorage independent cells, like I spoke just now about the suspension cells, they can grow floating in the media, like your HEK, your K562. The K562 are lymphocytes, HEK are your embryonic kidney cells. So you can see here how they look. Your anchorage dependent, they will attach on the surface and then they will spread like this, and they will be two-dimensional in your monolayer. But here you see your suspension cells, they are like spherical to take the maximum volume and surface area. Then they are all floating in the media. You can see few of them are in, like at lower place, they sink and settle. Few of them are at a different plane, so they keep floating. So these are all your questions which will help you in your assignment. I hope if you have any doubts or if you need any additional material for this, you can always, put up queries on the portal. Uh, if you want more details, we will give you that. So if anybody has any doubts, you can ask me right now. Anyone? Okay, someone answered here. When you're small. Okay, good. But as I said, you can, talk in your mic, because I won't be checking this when I'm in presentation mode. Okay, if anyone has any questions, you can ask me or you can leave it in your query sections. So if there are no questions, we,
I'm having a doubt.
yeah, please tell me.
What will be the question pattern for this exam, ma'am?
So the question patterns you can look for the previous years. You can check there. It will be available over there.
Okay, ma'am. So is it like for 15 to 2 or 100 one words will be given? So that's the case.
So that exactly I'm not sure, that the professors will decide. So you can put up a query there on the portal. Somebody from the support group will answer your query.
Okay, ma'am. Thank you.
Yeah. Okay. Okay. If there are no more questions, I think we can close the sessions. Thank you for participating and answering few questions at least. Thank you. Thank you, Sakib, Shahid, Kumaran, Himakshi. Okay. Bye.